Assalamualaikum. My name is Saida binti Muhammad Lia and my check number is S51386. I am from Bachelor of Applied Science Histories and my supervisor is Dr. Sharifah Nur Elia and I'm going to present a review on probiotics from the egg surface of sturgeon.
This is my content. It includes introduction, method, result, and conclusion. First is introduction. Sturgeon fish can be found in the subarctic and temperate region in rivers, lake, estuaries, and coastline. It comes from family of Achepanseridae and order of Achepanseriformes. The egg structure of sturgeon is complex and an envelope structure different with other fish group. It has a global demand and the egg's quality is influenced by the aquaculture technique and environmental condition. Little is known about the bacterial flora present on the outer surface of sturgeon eggs. Eggs in incubator for aquaculture will be more exposed to bacteria than most eggs in nature. Microbial communities were clustered by egg surface developmental stages regardless of flow regime. Probiotics are common bacteria, for example lactic acid producing bacteria are used widely in terrestrial animals. Probiotics should be tested for their inhibitory activity against targeted pathogen and tested their protection of host when challenged with pathogen. The objective of the study is to review the study about the presence of probiotic in the egg surface of sturgeon. For the problem statement, there is still lack of research about probiotic on the egg surface of sturgeon, what type of probiotic that could present on the egg surface of sturgeon, and how this probiotic could help in preventing fish disease. It has been a steady increase in the number of bacterial species associated with fish disease. Next is methodology. I use searching, screening, filter, organize, and report. For searching, I find the article at Google Scholar, ResearchGate, Scopus, Sendera, and NCBI. I use keyword sturgeon, egg surface, and probiotic, and the article will be saved on the file in desktop. Then I screening the title, abstract, and the result first to identify if it fulfills the inclusion criteria, and the paper must be in Bahasa Melayu and English. Then I filter the journal and exclusion criteria will be removed and it will be done manually by open all the journal one by one. Then I arrange the journal by author's name and year, student species, method and outcomes, and I use Prisma guideline to this review paper process. 16 literature study has been chosen, but only 2 literature study have been identified relevant with inclusion criteria. Another 14 were removed, and those 2 literature study are about egg surface on sturgeon. Next is result. This is the type of probiotic that present. Both of study were using the same species of sturgeon, which are lake sturgeon, Achepensa fulvescens. Fujimoto et al. 2018 has suggested a potential use of Pseudomonas species C22 for probiotic application as a tool to inhibit the proliferation of potential fish pathogen on the egg surface of certain fish species. On the study of Fujimoto et al. 2020, they have identified some of probiotic that present in their study, which are Archidovorax species and Massilia species. Both genera have a potential in use of probiotic in aquaculture. Next is method that used to analyze the data. Only one same method that has been used by 2 studies, which is using 16S rRNA gene sequences. In Fujimoto et al. 2018 studies, the presence of Pseudomonas species C22 was detected on the early stage of 8 developmental stages, but other Pseudomonas species were also detected in the late stage. For study of Fujimoto et al. 2020, they used terminal restriction fragment length polymorphism TRFLP technique has been used to compare microbial community composition across environmental gradient, location, and times. The total of 47 genera were identified, and the genera Archidovorax and Massilia were abundant in day 1 and day 4 of 8 developmental stages. Next is growth inhibition from antagonist interaction. Fujimoto et al. 2018 has done an antagonist interaction between 25 identified isolates in order to know which strains are pathogenic to the host and look for potential probiotic application. And they have found that Pseudomonas species were the most aggressive of all isolates tested. The table shows the 4 species that are strong aggressive strain compared to other isolates. Next is species with high relative abundance. The table shows that the highest and the lowest relative abundance of microbial on lake sturgeon egg surface at 2 factors and 2 developmental stage, which is day 1 and day 4. Genus Acidovorax and genus Massilia stated as the highest relative abundance in treated water, while genera Flavobacterium and Rahimera are more dominant in untreated water. For the conclusion, from the article review of Fujimoto et al. 2018 and Fujimoto et al. 2020, they suggested a potential use of Pseudomonas species C22, Acidovorax species, and Massilia species as probiotic. Many methods that has been used to identify the sample and several factors indicate the presence of probiotic on the egg surface of sturgeon. Water temperature had an effect on microbial community composition. Antagonist interaction indicate that there is potential of microbe to protect fish eggs against colonization and proliferation of pathogenic bacteria. There are still many types of microbe that need to find to guide further study on identification of microbe for probiotic egg treatment, as probiotic bacteria have been shown to be effective biological control agent in aquaculture. A bacterium is harmful to aquatic animal, but they can also bring a benefit to other species as probiotic. This is my references. That's all from me. Thank you.